sorpresa de la Facultad de Ingeniería y bienvenidos al seminario del Instituto de Ingeniería. Y les presento a Ehsan Shamlu. Ehsan es originario de Irán, estudió Ingeniería Química en la Universidad de Teherán, una maestría en Ingeniería Química en Cal State University, en California, y está por terminar el doctorado en Ingeniería Química también en la Universidad de California en Irvine. Tiene una sólida formación en detección electroquímica, en microfluídica y experiencia en procesos de micro y nanofabricación. Y durante su formación ha acumulado conocimientos y experiencias en proyectos que tienen inspiración biológica. En estos últimos 4 años ha trabajado bajo la supervisión del Dr. Mark Madhu, que también nos ha acompañado en otras ocasiones en la Universidad de Irvine, en California, desarrollando y optimizando una variedad de sensores electroquímicos basados en carbono, como el conjunto de electrodos interdigitados de carbono CIDA, por las iniciales, que son capaces de detectar una concentración de hasta un picomolar de un analito objetivo en un medio acuoso. Actualmente esta plataforma se utiliza para desarrollar dispositivos de diagnóstico inmediato, que en inglés tiene las iniciales POC, Point of Care Devices, que permiten hacer identificación de bacterias y pruebas de susceptibilidad a antibióticos en menos de 3 horas. Y hoy nos va a platicar Ehsan sobre un sensor olfativo. Y entonces, pues, démosle la bienvenida. Gracias. Thank you very much.
Thank you, everyone. Good afternoon, and welcome to this seminar. My name is Ehsan Shamlu. I'm from University of California, last year PhD student working in mostly electrochemical sensing, new material development, and I'm working at BioMEMS lab, UCI BioMEMS lab. I feel I'm very lucky that I landed for my graduate study in this lab, mostly because we have a diverse lab. We have 30 plus students in the lab, which we have more people from China, India, Mexico, Iran, Germany than actual Americans. So every day I have this extra motivation to actually make a trip to the lab to see these people from all around the world. It is quite a unique experience. Aside from that, BioMEMS lab is an interdisciplinary lab, which means that we work in different areas. We work in material development. We are specifically interested in carbon materials and developing carbon composites that they have enhanced mechanical and electrical properties. And basically, carbon is forming the backbone of whatever we do in the lab. I'm going to explain this, and pretty much everywhere on these slides, you see like a trace of carbon. The next thing we do is electrochemical sensors based on carbon material. Why we use carbon material? Because, first of all, carbon has a very interesting property. It has a very wide stability window. It means that you can use carbon in a solution which has like a wide range of pH without actually corroding or destroying the material itself, right? And we also can modify properties of carbon. We can create composites which are doped with electron donors, such as nitrogen, the same way that we dope, for example, silicon. Silicon itself, inherent silicon, doesn't have any interesting properties in terms of electrical properties, but when you dope it with elements from group 3 and 5, that becomes that superior silicon that we talk about, right? So we can composite, we can create carbon-based material that are doped, and on the surface they have, for example, nitrogen groups. These nitrogen groups they usually have an extra, nitrogen is from group 5, so it has an extra dangling electron. So that extra electron kind of facilitates the electron transfer when you're using it for the detection purposes. Other than that, carbon itself is kind of sluggish for electrochemical and detection purposes. That's why we are interested in doping the carbon. The next area that we work on is microfluidics. What you see here is a segment of a CD-like microfluidic device. In this device, you load all the chemicals and reagents on the CD, and then you spin the CD, right? You create like this micro centrifugal force, and you can process multiple samples at the same time. And using that force, you are basically evading any sort of interfacing with the air or a carrying reagent, which is very beneficial. Of course, it has its own disadvantages. I'm not going to talk about much about this. It's more about the lab background and what we do in the lab. Then we use a combination of these two to create something called point-of-care diagnostic systems. What's a point-of-care diagnostic system? This is pretty much what I'm going through the slide, a combination of number 2 and number 5. A point-of-care device is a cheap device which is aimed for diagnostic purposes. It is supposed to be cheap. It's supposed to be affordable. It is supposed to be easy to process and use. Why should it be cheap? Because we are aiming these for developing countries, India, like Iran, Pakistan, places people don't have money. $200 is probably 6 months worth of income of an Indian family. So no one is going to pay $200 to run a detection, a diagnostic, on the blood or urine or anything. So that is the goal of this, to make this globally available. And another interesting part that we recently got involved into it came up after the ECS conference in Seattle. The idea of growing stem cells in a certain direction, is very insightful. And why is that? Because we have many people that they are suffering from spinal cord injuries. What does that mean? That means that there is a segment cut in your spinal cord and you cannot walk. Basically, neurons cannot communicate with each other. People can grow stem cells in a bulk, in a random fashion, but they cannot grow it in a certain direction. They cannot force the stem cells to grow from point A to point B and hopefully be able to create that junction, that bridge between two broken points and basically facilitate the signal transfer. So that's what we are trying to do. As you can see, there is a randomly patterned carbon material. Again, carbon is extremely biocompatible. Anything bio you put on it, it's going to survive. It's going to, evolve. And on the right side, you can see like a very linear fashion. And if you see as a big picture, this is a small SEM segment of a picture, cherry pick the stuff. But if you look at the big picture, you can see like everything in the second on the right side is aligned with the fibers. So we are working on that. Right now we are working on mice stem cells. And the last part that we are interested in working in BioMEM's lab is biomimetics. This is the trichome from a bed bug. Historically, we know that people put leaf beans around their bed to prevent these bed bugs getting actually to the bed. And when you take SEM images of these leaves, you realize that there are these small hooks that you can see here. They actually penetrate into the legs, to the trichomes of these bugs, and basically trap them and kill them. So we are trying to mimic the same sort of feature using carbon mats, right, to be able to trap these bed bugs. For the sake of this presentation, I'm mostly focusing on electrochemical and point-of-care detection and why we are interested in those stuff. In 2015, I think I was, again, very lucky to meet this guy for like a brief 15-minute period. I was walking from that was my first year of research at UCI, and I was walking from the parking structure to my lab, and I happened to meet this guy. And he is a retired urologist from UCLA, and he asked me 2 questions. The first question was, what is the biggest threat that you can forecast, and what is causing it? Well, at the time, I was like, maybe Iran, nuclear weapon, Donald Trump was not the president at the time, so I couldn't tell him like Donald Trump. So I was like, I'm not going to give you that answer. He started already smiling like, you don't have my answer. And then he followed up with second question, and he asked me, what hasn't changed over the past 100 years? And I think for any researcher, PhD student, anyone who wants to get into the field of research, these are the type of questions that you want to ask yourself. You don't want to get into academia to just publish papers. You want to make that dent. And before that, you have to ask these kind of questions from yourself. What are you investing on? In a matter of seconds, I went over everything, phone, traveling, communication, fashion. And I was like, dude, like everything has changed over the past 100 years, except one thing. And that was his answer. If you know what is this, you probably know the answer, right? On the left side, you see a culture dish. What is that? That is a sample from a pathogenic sample, which is grown on a culture media. Basically, you have a sample which you know for sure there are like pathogens in that, and you grow that pathogen. And you see different colonies from the shape, from the size, from the color. You can see these are different bacteria. And then you take that sample, put it in a different sample, different petri dish with a different food, and try to grow those and expose to these small discs. These discs are basically antibiotics, right? So what happens when you go to the doctor? When you go to a doctor, they take you a sample, right? And the doctor is seeing some stuff. He's like, okay, you have these symptoms. You probably have this bacteria. And he's going to take that sample to the laboratory. It is going to take them 72 hours, 48 to 72 hours, to figure out actually what exactly is that antibiotic, is that bacteria, and what is the most effective antibiotic on that bacteria, right? So this process from getting here to the end of this, it takes almost 3 days, right? And meanwhile, on the same day, while you're sitting at the office, he's going to prescribe you, based on an educated guess, take this antibiotic. There is a high chance, there is a low chance, you never know. The thing is, the doctor, when he's prescribing you the antibiotic, he doesn't know what kind of resistance you have in your body, right? So he tells you at the end, within 3 days, if you are not feeling any better, come back to the office, because at the end of 3 days, he has the answer for the best antibiotic, right? So he's basically gambling on your health because he doesn't have an immediate solution. Statistically, there is only 23% that he gives you the perfect antibiotic at the perfect dose. And this is contributing to more than 80% antibiotic resistance in countries like Iran, India, Spain, Pakistan, and all those countries that they don't have a rigorous antibiotic regulation system, right? So you can see this is the problem. The detection process is a problem. As of now, I'm talking right now, there are only 4 FDA-approved automated systems that they do this kind of diagnostic. They are not significantly cutting the time, maybe cutting it to 1.5 days, but it is not still within that window, 1, 2, or 3 hours while you are sitting in the doctor's office. So still, the doctor has to gamble on your health. And there is one more fun fact, I would say not so much fun, that there has been absolutely no new class of antibiotics developed since 1983. And when you get antibiotic resistance, you don't develop that to a certain type of antibiotic, you develop that resistance to a class of antibiotic. And the conclusion is that there are only 4 antibiotics left, according to CDC, that we have more than 95% liability worldwide to be able to treat basically pathogenic sample using those. Everything else, we have some sort of resistance. And this is very scary. A lot of companies then don't even get into developing these antibiotics because it is not financially sound for them. They're going to lose the patent because antibiotic resistance is going to grow way before they lose the patent. So I'm going to introduce some terminology, and then I'm going to dive through the essence of what we do in the lab and what I do for my PhD thesis in the lab. First of all is the detection protocol. Generally for detection protocol, any sort of detection that you do, there is a surface, you have an electrode. It can be gold, it can be a porous material, it can be cellulose nitrate. You have a substrate that you have like a specific type of capture probe attached to that surface, right? And then what you do, you add the target to that sample, right? So that is that target that you're after, that bacteria, genomic data, whatever you're after is right now attached to the surface. Right now at this point, you can use techniques, which we call them probeless techniques, to detect the bacteria. What are those probeless techniques? For example, you can use SPIR, you can use you can measure the weight of this substrate before and after. You can measure the resonance frequency of the substrate before and after to basically determine the amount of target in your sample, right? When we talk about optical and electrochemical detection sensors schemes, it is good to know that like all of those biological samples, let's say most of those, they are neither optically active nor electrochemically active. It means that they are not going to emit any light and they're not going to exchange electrons, right? So what would happen here is you have to add another segment to it, which we call a detector probe. If you look at this yellow thingy on the top, we call that a fluorophore. It means that you shine some sort of laser light excitation source, and because of the Stokes shift, you get a different wavelength at a different intensity coming out of your substrate. And based on this light intensity that you get from the substrate, you can quantify what you have actually on your electron. Now, if you want to do electrochemical detection, you have to facilitate that electron exchange. You usually add a catalyst, and then you have like another chemical which is electrochemically active, and then you measure the electron exchange properties of the substrate, and that is how you amperometrically detect the concentration of pathogen in your sample. There are three more terms in this realm that I want to talk about. Sensitivity is basically the slope of the dynamic range of your system. You don't want your system to be very sensitive because it's going to reduce your dynamic range. Basically, you want to go over the whole broad range, and you don't want to be very flat because in that case, you won't be able to get decent signal in your dynamic range. Selectivity is again what it is that your system, how good it can distinguish between target A, target B, and target C. It is important to have a good level of selectivity. And also lower limit of detection, statistically it's defined as 3 standard deviation above the background. Basically what you do, you start running the experiment at a known concentration of target, and you create this black curve. And 3.29 over your standard deviation means that it is going to define your dynamic range. So your assay, your system is going to work anything above this line. This is a very important topic. It actually defines a very important differentiation in the realm of detection. We have two types of detection schemes. We have a genotypic type. You have a cell, you break it down, you take DNA, RNA out, you extract genomic data, and then you try to put that on that electrode that we had, and based on the genomic data, you detect, what is in the system. We also have phenotypic systems. You look at the shape. If you remember, each bacteria colony had like a different shape, different configuration. Based on that, we call that phenotypic. The advantage of genotypic is that it is very fast. Within 2 hours, you can determine what it is. But the instrument can cost up to $500,000. And it is not a small, usually probably I would say about half of this table. An experiment itself is about $200. So that is definitely not suitable for countries that they suffer from financial restrictions. But at the same time, it is very precision. If you have the right capture probe, it's going to definitely give you that answer, no question. But at the same time, if you have the right capture probe, if there is any mutation because you don't have the right capture probe for that newly emerged mutation, you won't be able to detect it. Last year, I think they had 2 people died at UCLA Hospital Medical Center because they are suffering from newly emerging bacteria. But on the other side, phenotypic is pretty long. It can take 2 days, 3 days, automated maybe a day. On the downside, when you're culturing chemicals, culturing pathogens, you can culture extremely dangerous pathogens on your culture plates. And imagine if something goes wrong with those culture plates, right? So this is the comparison of these two systems. We also, for the sake of electrochemical techniques that I'm talking about, we talk about redox reaction. And redox reaction is a reaction where an oxidizing agent receives an electron and becomes a reducing agent at a certain potential, right? And these are defined here. So in an amperemetric technique, we measure the rate of exchange of these electrons here. This is what we are interested in. We know electron current is defined as the rate of exchange of electron per time, right? So now the big question is, we had some terminologies here. We had the principle of detection. We know how to assemble this stuff. The big question is, can we detect anything without actually amplifying the signal, without amplifying the source? If you have, going back to this reaction, if I have one molecule, one electron coming out of it, is there a chance for me to detect that pathogen as a reliable level, right? So here's the game that is coming on. I want to go through some theories, and then we'll see what's our solution for this problem. Imagine again you have your capture probes here on your gold substrate, right? And then you now add your target molecules, let's say DNA, RNA, proteins, to the same complex. There are these physical sites on the surface available, right? Statistically, some of these targets are going to bind, form like complexes here. Some of them are not, and some of them are not, you know, hybridized to the capture probes. This reaction, this adsorption, let's call it adsorption-desorption reaction, the surface reaction is governed by this equation. The forward reaction is governed by something we call it K-adsorption. This is the rate of adsorption of target to these capture probes, and the KD is rates of coefficient rate of desorption. Basically, once we get that to that equilibrium, these guys also are going to leave the surface for the same reason that they are basically getting connected to the surface. Then we have an interesting topic called Langmuir approximation theory. He assumes that the total number of sites are equal to the ones that are hybridized and the ones that are not hybridized, right? And also he defines variable theta as the number of hybridized sites to the overall sites. And then when you combine that with the above equation, you get this equation. It's a very simple equation. It's the governing equation for the Langmuir approximation. Assume that you're at the equilibrium. You gave some time to the cell. It is getting to the equilibrium. At the equilibrium, we know that this is going to, d theta over dt is going to get to 0. You derive equation number 2. This is the fundamental equation that we are going to talk about. You can also derive the time-dependent equation. I don't care about this. I'm just going to focus on this, why this is important, right? Let's have two examples. I'm talking about a 50 nanomolar concentration of analyte. If you have the initial concentration of your target is 50 nanomolar, if you add a 50 nanomolar analyte to your solution, to your cell, detection cell, if you put these numbers with this K adsorption, which is a practical number, if you put it back here in, where is the equation? In this equation, you get this number. It means that out of every 2 sites, one of them is taken. But is 50 nanomolar, is it really a good concentration for a detection system to be able to give you reliable data? No, not really. If the concentration of the pathogen in your body is at this level, you're already dying. It is like zero chance almost, say, to basically help you, right? So what you want to do, you want to reduce this concentration. The practical concentration for a competitive system is 1 femtomolar. Let's put this 1 femtomolar with the same K adsorption to the same equation. It gives you this number. It means that out of 100 million sites that you have on the cell, if you're lucky, you're going to get 2 adsorbed on the surface. And if you can get such a system that has that kind of efficiency, man, you're lucky. I would say go out, buy the lotto, you're going to win it, right? This is not going to happen. So we need a strategy to get from this to this. How can we tackle this problem? We have two different solutions so far. There are two distinct solutions for this problem. If I want to split those, I would split them to the assay level, to the chemical level amplification. People add, for example, catalyst to the detector and try to amplify that signal. People add different sort of chemical to basically somehow chemically amplify the signal. But for the sake of this seminar, I'm talking about the sensor level amplification. What does that mean is that you add some sort of physical manipulation on your sensor to be able to amplify the signal. And everything relies pretty much on these equations that we wrote at the beginning, right? I'm going to talk about why this is crucial. When we talk about electrochemical sensors, there are three terms. We are talking about mass transport phenomena, right? You have a diffusion property. You have a diffusion on your electrode, which is because of the concentration gradient happening in the system, right? You have something called migration. Because your system is a charged system, you have a working electrode which is charged. You deal with migration properties, and you also have something called, we call it convection, because you have some sort of mixing, usually temperature gradient. It basically pushes the molecules to move from one stage to the other one. And you also might have some sort of reaction. Let's simplify this equation as far as it goes and see what we can get. So typically we assume that there is no reaction, and typically we don't want to have migration in the system. How can we kill that migration? We add some kind of background salt to be able to kill that electric field in the solution. KCl, PBS, there are different form of salts. And on top of that, we can also not mix the solution. We can design a microfluidic chamber, put it on our sensor, right? And basically eliminate the velocity vector. And at this point, you get this simplified reaction, which is the Fick's first law, basically. Actually, Fick's second law. But it is important to understand every single of these terms here. We are talking about a gradient term. We are talking about the diffusion in this problem, right? What is the diffusion layer? If you have if you assume that there is, in this room, there is no convection, there is none of these air fans is working, and if you assume that there is a temperature is uniform all over the place, if you spray a perfume in this corner, if you stay here probably like 10 years, you might be able to smell it on the other corner of the room. So it is an extremely slow process, which is caused by the concentration gradient of a species that you're talking about. So I simplified the system, and I'm talking about this. Now I'm going to go back and look at this problem, the equation that I had on the top side. So we said we want to amplify the signal somehow. How can we amplify the signal? A typical electrochemical cell has 3 working electrodes. It has a working electrode. Ignore the other 2, work reference and a counter. There is the working electrode, that the whole thing happening on it, right? And this is pretty much a reaction in this direction that is happening. You have a species, it receives an electron, it becomes another species. At this configuration, if I have 1 O, I can give it 1 electron and I can generate 1 O, 1 R, right? So I only consume 1 electron per unit of second time, right? What would happen if I consume this and push it back and feed back the system, right? So at the same time that you're consuming, you will have another electrode, again, creating the same species and putting it back into the system. So instead of using it only once, you can use it multiple times. Instead of creating 1 electron or using 1 electron, you can create or use more than 1 electron. And this is something that we call it redox amplification. So the question is, how can we design the system? How can we have the best performance out of the system? Before that, you need to understand also the concept of the diffusion layer. Assume that in this case, so typically when you run an electrochemical detection and amperometric detection, you apply a linear sweep. You say, for example, to your apparatus to go from VA, V1, let's say 0 volts, to 600 millivolts at this step, at this ramp, right? And what is happening, assuming that this is the surface of your electrode and this is the depth of your solution, right? This is the surface of electrode and this is the depth of my microfluidic channel. As you start applying the potential, you start consuming the reagent. This is concentration of the reagent. This is the concentration of the product. At the beginning, it is 0. At the beginning, it is equivalent to 1 when it's normalized. And you start consuming it, and regenerating the R to the point that you see eventually this cycle forms, right? So when you're running it from 0, you can see it starts from here and forms this nice peak up there. And what is the translation of that peak? It means that when you apply this potential, you start consuming your reactant on the surface of the electrode, right? At some point, you have used so much of these chemicals. You see, this distance is getting larger and larger, that the diffusion rate, I mentioned it is a very slow process. The diffusion rate from the bulk is less than your consumption on the surface of the electrode. So at that point, you see that your signal intensity drops and you cannot generate as much current, right? So once you have the reverse of this cycle, you will see like the reverse of this signal. So this is pretty much how we interpret diffusion limited current in a system, because at this point, the diffusion from the bulk cannot compete with the rate of consumption on the surface of your electrode. So again, we talked about this. How can we amplify the signal? Instead of one electrode, we can have 2 electrodes sitting next to each other. This is a demonstration of a very thin layer of gold electrodes. This is about 20 to 50 nanometer of gold electrode on a substrate. You can see the bulk is diffusing to the surface of the electrodes, forming the reaction, cycling between the 2 electrodes, and going back to the bulk solution, right? Now I want you to use your imagination. I said this is about the height of these, because these are made from gold. Because of the technical limitation that we have, we cannot create these high aspect ratio structures. These are only 20 to 50 nanometer, very thin, right? Now I want you to use your imagination and increase this height. What would happen? Instead of one cycling, you will have multiple cycling, right? And this is a trick, exactly what we were talking about. We want to create high aspect ratio electrodes to increase the number of cycling occurring between 2 working electrodes. And this is the results. So as you can see, these electrodes are probably 1 micron height, made from carbon, extremely stable at the pH of interest for biological detection, which is about 7. And it also has a high aspect ratio. It means that instead of having one of these and getting only 1 or 2 electrons out of the system, you can get multiple of these electrons out of the system. And now I want to have a quick comparison between these 2 systems. Why we are interested to make these from carbon. Typically, the fabrication process starts with the silicon wafer, and then you coat a photoresist on a silicon wafer. At that point, you do the lithography, and you create like a pattern on the surface of your electrode, right? On the surface of your wafer. Now, you put this photoresist, pattern photoresist, into the furnace. In the absence of oxygen, you pyrolyze it, and you get some sort of carbon depending on the temperature that you're setting, right? But this system, this side, on the metal side, it is still going on. I still need to deposit chromium because gold or platinum is not going to stick to SiO2. And then I still, after that, I need to add the layer of gold. And after that, I need to remove whatever I don't want. These 2 steps, they are performed in high vacuum processes, E-beam, thermal metal evaporation, and these are extremely costly. plus on top of that you're using gold and platinum. This whole system is much more expensive. So this is why we prefer to have carbon-based interdigitated electrode arrays to amplify the signal rather than using the metal ones. We have solid reasons why we should go into that path, because if you remember at the beginning I mentioned about point-of-care diagnostic. This chip has to be less than $1. Right now it is about 60 cents, which is right on the price point. People in India, maybe with subsidization, can use it. Maybe if we take this to industrial facility to fabricate those, maybe they can use it, right? Now I want you guys to look at these two signals. I mentioned about this signal. You start applying the potential, you start consuming the electrochemical species, and at this point you're out of the electrochemical species on the surface of your electrode, and you see that your signal decays, and then it goes back. In the opposite side, when I have two electrodes at this configuration, you see that the signal goes up and plateaus out. It stays flat as long as I'm applying the potential. This is what we call it a steady situation. It doesn't matter if I'm measuring at this potential, I'm on a flat line. This is the beauty of the system, right? But here, post-processing is kind of difficult. The other thing I want you to pay attention is that the maximum current here is about, let's say, 14, 15, 12, whatever this number is. What is compared to this number? It is 15 times at least higher than that. So that means that instead of having one electron exchange, I had 15 electron exchange. It means that I created that redox amplification that I needed to get that to 1 femtomolar, right? So I'm one step closer to that. Now, the interesting part about this thing is that when you're looking at these IDAs, interdigitated electrode arrays, there are 3 parameters that you can manipulate. There is the width, there is the gap, there is the height of these electrodes. We change all of these variables and try to see what is the effect of each of those variables, right? For instance, we change the width of the electrode from 0.1 micron, 100 nanometer, to 10.1 micron. And you see that this is not linear, but kind of like saturating at 10.1. And this makes sense because if it is at 10.1, I'm almost getting to a macro electrode. And this is like a common sense for an electrochemist, of course. If you have a higher surface area, because electron exchange are surface properties, surface phenomena. If you have a higher surface area, you're naturally going to have higher current density. And if I change the height of the electrode, because I'm basically having more of those redox cycling happening between the electrodes, I'm also amplifying the signal, right? So the current goes, as I increase the height, current amplifies. But the interesting part is here. If I bring these electrodes as close as possible to each other, I can see almost a 50-folds amplification on the signal. So the point is to bring these electrodes into each other's diffusion layer. Once they are overlapped, once they are placed, if I go back to this slide, once these two are maximum, they have the maximum interference, you have the maximum signal out of that. And for that reason, you want to design your sensor in a way that the fusion layers are overlapping each other. Going back to this signal, what is this telling me? Is that I need to bring these two electrodes as close as possible to each other. But is it possible? With the scheme that we have, with the lithography, the process that I showed you, it is not possible really to resolve something that high aspect ratio, it has a very small gap. At some point, these electrodes are going to merge on the bottom and everything is going to short out. So that is lithography. With a conventional lithography is not going to happen. And I am not interested in using Intel technology to make 7 nanometer gap because that is nonsense. I cannot spend $70 for a chip. The whole idea was to make this chip. Our solution is a nano gap. How do we make these? We make the same electrode lithographically process. We lay a nanofiber on the carbon electrode, right? Then we passivate the whole system, the whole fiber, and the microbands with silicon dioxide, because we want the electrochemistry only happen at this gap, right? And at this point, we put the electrode inside a vacuum or nitrogen chamber, and then apply potential, and because of the Joule heating, it heats up. Silicon dioxide, you see the small ball here, sublimates, and it creates like a small crack here. And that's all I need to do for extreme redox amplification in the nano gap. This gap is about 27 nanometer. It is still way bigger than the dimension of the diffusion layer. Sorry, smaller than the diffusion layer, but the whole geometry is going to work for me because right now I am into that diffusion layer. Unfortunately, I don't have any experimental data because we got this working almost 10 days ago last week. So this was a lucky image that we got, and by the time I go back to California, I have to start running experiment in nano gaps and kind of quantify this. But on the dirty runs that we tried in the lab, because once you create these gaps, you're like, oh my God, is this going to really work? We did the dirty runs. On the dirty runs, it is working, but we don't have any presentable data on these gaps. These are some interesting features that we came across during the fabrication of the nano gaps. So this is the fabrication process. Let's assume that you have this microfiber here sitting on your electrode, and you apply the potential here, it heats up, and it melts down and creates this gap. Do you see this effect in the back? This is because of the high temperature at this gap. It is so high that it is melting the SiO2 on the surface of your silicon wafer. So this is a very interesting phenomenon. And if you're paying attention here on these corners, you can see sublimation residues of SiO2. The second thing is that if you do the same process in in air, you probably see something like this. The glass silicon dioxide, because of the convection properties, because you change the convection properties of the system, the glass cannot sublimate, and it only melts down and breaches. And if you suddenly increase the voltage, you create this funky shape, which basically it's like I apply 60 volts immediately to the microfiber, and it just bursts everything. So these are interesting phenomena that we observed on the nanofiber gaps. And the point is here is the next level of sensing. What can we do with this? The problem with electrochemical sensors is how you can amplify the signal. So we can potentially amplify the signal. How many of these can you put like in a practical footprint? The sensor that I can put like, let's say about 1,000 of these nano gaps is going to be 9 millimeter by 9 millimeter. So it's going to be pretty small. Compared to the Enos that they usually have like 32 sensing segments, we can have technically thousands of these on the nano gaps. And hopefully, with the finding of the right membrane, we could be able to get to that actual olfactory system that replicates like a dog or a bug, the way that they smell that stuff. We are trying to populate as many as probes possible on a small surface to be able to amplify the signal and statistically get the right signal. I want to say thank you to all these folks. These are people from my lab. Dr. Mark Madhu, he's my advisor. He's a great PI, greatest PI one can ask for. And every single one in this image has been working with me in different areas. And also we have a postdoc in the lab, Lola the postdoc. So she's also a help to many of these students. Thank you. And I'm open to any question.
Thank you very much, Ethan. Thank you very much for your talk. Si hubiera alguna pregunta que quisieran hacer a Ehsan Shamlu.
Yes.
Yeah. So you use a photolithography to make the carbon-like electrodes, and then how do you make like the fiber? How do you make it like that small? Because if your final gap is like 24 nanometers, that fiber should be like 100 maybe in diameter.
100 nanometer or 100 micrometer?
100 nanometers in diameter.
Well, so that part is kind of like lucky how we make it, right? So we usually have the same material, the same polymer, SU8. It is a photosensitive polymer. And we put it in a syringe, and we create a micro droplet at the tip of the syringe. And we have a glass needle coming it, touching it, create like a small line, and then moving it, putting it down on the fiber, right? So this is how it is. And this is not an automated process. We get a lot of lucky things, moments to do it. But once the lab humidity is fine, if the same person is working on it, if the temperature is fine, humidity is fine, if everything is fine, you should be able to get to that specific diameter that you want. The problem here is, if you want to get a very thin fiber, if the distance between these two pads is wide, the fiber is going to sag down and it's going to touch the substrate. So within that process, we have to optimize these gaps. So on an electrode, we have, I'm sorry, we have multiple designs, but we have anything from 1 micron gap to 200 micrometer gap. And depending on which gap you are using, we can adjust, we can engineer the diameter of that fiber. So it is kind of a lucky process, and it also depends on which gap you're using in the fabrication process.
All right, thank you.
Thank you for your talk.
Thank you.
When you talk about selectivity, you say a good level of selectivity, but if you could talk about more. But how could deal with the lack of selectivity if you want to identify several microorganisms or bacteria? And how could we improve this selectivity in your technique?
Okay, so
Thank you.
Thank you for your question. So the selectivity mostly comes from the hybridization chamber, right? So what usually happens is that you have these specific capture probes in the hybridization chamber, right? Let's say, for example, talk about UTI, urinary tract infection. You are checking for 14 different bacteria. And at the same time, you want to have the positive and negative control. So you need to have 16 of these on your cartridge. 16 different capture probes, your positive and negative control, right? Once you have that hybridization process done, right, we use a technique called flow injection analysis. What is that? Is basically you have 16 of these chambers and you have a detection cell with your electrode, right? You inject the solution from the first hybridization chamber over your electrode, you get the signal. That is from your first bacteria, right? And then you wash that electrode with a buffer solution. Then inject the second one from the second. And sequentially, you introduce your analyte over the electrode, right? And for each of those specific, you get that specific signal. And then you take it to the calibration chart, and then you figure out which one is which one. So for the sake of selectivity, in electrochemical sensing, the best way is something called, we call it flow injection analysis, right? In optical detection, they do different techniques. For example, they have these beads, microbeads, 1 to 50 micrometer in diameter, which at the core of that bead, they have different fluorescence, RGB, right? Actually, they have a combination of RGB. These are standards, colors. And then they have the capture probes, instead of having it on the electrode, on the flat, they put the capture probes on those microbeads, right? So when they scan for the intensity of the light emitting from that, they are checking for multiple wavelengths. First, they check for the intensity of the microbead. They say, for example, this combination has this capture probe on it, and it's also emitting this light. So that's how they adjust the selectivity. For that, there are multiple patents, for example, for the microbead techniques. The best one, the most improved one, I think it's from an American company called Nanogen, if I'm not wrong. They have the most complex, but 100% working, and you can multiplex, I think, up to 96 different things at the same time. So there are different techniques for how you want to amplify it, but again, it has to do with how you define these capture probes in your system. For example, like what I said for EChem, we prefer to have a flow injection analysis, which is pretty simple, and you just need to modify your microfluidics.
¿Alguna otra pregunta?
Hello. My question is in the same line. I wonder what kind of compounds is the, how do you call them? Capture probe. What kind of compounds?
Oh, capture probes? These are different depending on what you're going to detect. It can be a protein if you are doing something like ELISA. But in my case, for example, I want to talk about actually more about, maybe if I give you an idea about this, it's going to simplify the problem. One level of amplification that we use in my assay is that I take the bacteria, we lyse it, right? What does that mean? It means that I shear the cell wall and I take the DNA and RNA out and then I stabilize it, right? We know that 16S RNA in bacteria is a reserved RNA. It means that it doesn't mutate from one type to the other one of the same lineage, right? For example, if you're going for after to detect E. coli, it doesn't matter what mutation you're going to. If you extract that 16S RNA from E. coli, it's going to be the same within that lineage, right? So I extract that 16S RNA, and then I order to the company, I know what is the configuration of that 16S RNA. I order the proper capture probe to that 16S RNA, right? So it depends what you're going after. In my case, again, it is an engineering capture probe, which is a single-strand DNA. One more thing to mention about is that this is another level of amplification, right? When you break the cell wall, for each cell that you get something between 10,000 to 100,000 16S RNA. What does that mean? You're amplifying your signal, right? You're increasing that odd. If you have one bacteria, you already have, like, let's say 10,000 16S RNA in your sample, and you're improving that probability function to get a better signal.
I understand. So my second question is, I work with bad odor air. And I read in your presentation that you were talking about odors detection, and I'm wondering what kind of probe can you use for improving the odor detection, for example, of ammonia or hydrogen sulfide, something like that. That this is also in my topic is interesting to measure odors.
So this is, again, the motivation for that was when I was looking at my dog, Lola, she was sniffing all over the lab, and I'm like, how can she distinguish between all of these stuff? And the first thing when you design an electrochemical sensor, the first thing that you can do is a gas sensor, right? For odors, for smells, you are mostly relying on a conductive material, which is basically selective to the ions from the bulk to the surface of your electrode. For example, how are these gas sensors working? You have two electrodes, right? You have a material here. Once you have a certain level of carbon monoxide, that's going to diffuse into the bulk of that gel, right, and create a short, and then it's going to buzz the thing. The same concept. I'm not getting into developing that chemical compounds For it, I'm mostly interested in putting so many probes on a small surface to be practical. At least at this point, for this purpose of my thesis, that is not a part of the game, hopefully, because that I know that is going to be another 5 years of work, and I don't have that time at this point. Yeah, but it is mostly to the knowledge of chemists and material developing people to develop selective material that you can at the same time create like nano droplets on certain regions. My task as of now is to create so many of these probes on a very small surface to be able to get that ion diffusion from the bulk to the surface of the electron. Yes.
Thanks, Esan. So the amplification is in the nano gap, right? And you were talking about also the interdigitated electrodes that amplify the signal, the redox signal. So do you think that you can get like a nano wall or something? I mean, in terms of the question that Oscar was doing, depends on the diameter of the fiber. It's going to be also I guess, the gap size, right? But maybe you can have a very small gap, even if the diameter of the fiber is larger. Then you have more redox amplification.
This is the thing. This is going to be an optimization problem, right? So I don't know if it's worth it to go that far down to overlap everything. I have to. My simulations are still running. I haven't touched like the experiments, but I am pretty sure smaller gaps at some point is going to give us a problem, because when you have the working electrode, and once you do the cycle, at the same time, you are accumulating a species on the tip of your working electrode. And if this is going on for a long, it's going to short the two electrons.
Or could you tunneling current?
Could I do it? What?
Like maybe there are tunneling current, like jumping?
Well, this is actually something funny that happens here. This is the last image, actually, I think, on the slideshow. This one. So this guy here, when we had this phenomena, this was working in a vacuum environment, right? When we had it, it was like an LED. So it brightened up so much, and like the whole like working area, we had like a bright LED into it. And once we were done with the experiment, we could see that this is a junction. This is sealed on the outside with SiO2, but the inside, we cannot kill the current. There is a 60 nanoamp current going all the time into it. As soon as you start.
Nano current is probably a tunneling current.
That is a tunneling current, right? So that is what is happening. But again, this is like, that's why I put this uninteresting stuff, because this can trigger some applications for it. But in this case, the SiO2 melted around the fiber, and you could see that tunneling effect of 60 nanoamp going constantly through the wire.
¿Tenemos alguna última pregunta para Esan?
Hi, thank you for your talk. We are working with detection of pathogens, but with molecular biology. And we also know that if you use 16S rRNA, you have, depending on the length of your probe, you have different selectivity. Because if you take a long segment of DNA, you have better selectivity in the database of the NCBI, so you can identify the microorganisms with the length.
That is correct. So 16S RNA, I think, has like about 1,500 nucleotides.
Exactly.
And that means that it is very like long and curly, and it is going to reduce the specificity and selectivity of your probe. There is a patent. I cannot disclose how I do that, but I can give you the hint. There is a paper published in 2013 by Colin C. Hofford from UCLA Medical Center. So after they lyse it, they use sodium hydroxide. They break down the segments of 16S RNA to the smaller segment, which increase the selectivity.
Okay.
Just search for amperometric detection of, actually I think that is on Jovi, you know, Jovi, right?
Yes.
Yeah, it is on Jovi. Just Google for Jovi electrochemical detection, Colin Halford, Halford. But that is a patented procedure, as far as I'm sure, in the United States.
Well, thank you very much for your talk again, Esan.
Thank you.
We give you an applause. Te damos un aplauso.
Thank you.
And we have a present to him. We have a present, Laurita. We have a certificate and a gift. I have here. Not precisely, not for you, for Esan.
I think I should come to this way.
It's a cup and a certificate.
Thank you, I appreciate it.
Thanks to
Thank
you.
you.
Thank you very much. Muchas gracias a todos.
Thanks. It is really exciting when you see people who are interested to know the next level of science. I myself get bored when I work in aerospace because it is kind of getting boring. But next level of science is something that you want to devote your time and energy to, because the next generation is always matter. Thank you for coming here.
Thank you again.